It's on. Well, it's on. Yeah. Okay. Cheers. Bye. Okay, year two. Welcome back in whatever shape or form that is currently taking. And welcome to the OV 2023 visual perception module. Important components of the first term content on this module pretty much continues some of the themes that we discussed in first year neural and visual biology. I mean, especially contributions of the eye, the retina, and the optic pathways to visual function. However, visual perception actually is a product of the human brain. And so to begin the module, we're going to begin with a little 4-lecture mini-series aimed at familiarizing you with the structural and functional organization of the brain. And this will be supplemented by an online tutorial session with a formative assessment associated with it in a few weeks' time. In fact, to really begin, I just want to start with a little reminder about some things that we covered during the first year neural and visual biology module. So, for example, you will have been told, and I hope you kind of recall, that there are two main classes of cell that are the constituents of the nervous system, both central and peripheral. These are the kind of building blocks of the nervous system, and they're called neurons or glia. You may also recall that neurons are the ones that generate electrical potentials, and that they consist of several components. So, for example, all neurons possess a cell body, otherwise known as a soma, and within the cell body is the nucleus of the cell, which contains its DNA. Coming out of the cell body of neurons are fine processes. Quite often there are several of them that come out of the cell body, and these are known as dendrites because they're kind of tree-like structures with branches on the end. And it's onto the dendrites of neurons that synapses from other neurons are applied and signal to that neuron. All neurons possess another process, and it's shown here. It's called an axon, and there's only ever one of these. And the axon travels some distance, and on the end of the axon is another branch-like structure. It's called an axon terminal, and on the end of those axon terminals are synapses which the neuron is able to apply to other cells. Sometimes they're neurons in the nervous system, and sometimes they're outside the nervous system. You may also recall that the other type of cell within the nervous system is called glial cells, and the word glia means nerve glue. And these cells play really important supporting roles for neurons. There's one over here on the right. It's called an oligodendrocyte. Oligo is few, dendro is branches, cyte cell. So this is a few branching cell, but the few branches it does have, it wraps repeatedly around axons to form these things, which are known as myelin sheaths. And the oligodendrocyte is the cell responsible for myelinating axons within the central nervous system. You may recall, of course, that impulses are conducted from the neuron cell body down the axon to its terminal synaptic branches here. And the function of the myelin is to increase the speed and also the reliability of impulse conduction. Oligodendrocytes are the myelinating cells in the central nervous system, or CNS. The counterpart in the peripheral nervous system, or PNS, is the Schwann cell. We know that these oligodendrocytes are engaged in really important roles because of demyelinating diseases that can affect the central nervous system, in which the oligodendrocytes are attacked. They strip their myelin off axons, and then impulse conduction ceases, and the person suffers from severe neurological deficits. On the left here is another type of glial cell. It's a very common cell within the central nervous system again, and it's called an astrocyte or star cell. Astrocytes also have processes on them, and they perform a variety of roles, mainly in mechanical support for neurons and for their axons, and also, as indicated here, forming perivascular end feet, which they wrap around capillaries in the central nervous system and form part of the blood-brain barrier. We also talked in general terms about the overall organisation of the nervous system, and I told you before that the nervous system comprises two main components: a central nervous system, which consists of the brain and the spinal cord, and a peripheral nervous system, which contains a whole bunch, thousands of individual nerves that contain either sensory nerve fibres that report information, for example, from receptors in the skin to the central nervous system, as indicated by this blue line and the arrow leading towards the CNS, or send motor commands from the central nervous system to, for example, the voluntary muscles to cause them to contract. So that's a motor output that comes from the central nervous system down motor nerves. And we also talked about how there is embedded within the nervous system an involuntary or automatic component known as the autonomic nervous system, indication of which is shown here. It was an example which I think I showed you last year about how you have receptors in the arterial walls of the big arteries that come out of your heart and detect and report the state of blood pressure, the pulse to the central nervous system, and how there are nerve fibres. These are not under voluntary control that come from the central nervous system and regulate the force of heart contraction in order to therefore regulate the strength of the pulse and hence your blood pressure. Okay, so that's kind of where we were at the end of last year, and now we're going to embark upon a rather more sort of serious investigation of the structural and functional organisation of the human brain. This first lecture is actually a slightly sort of gentle introduction to the human brain, and I just wanted to say there's a real sort of major upside to this. Indeed, you are really, really lucky to be listening to me today, telling you about the human brain, because the upside is that the human brain is undoubtedly our most exciting organ. I don't mean just because it contains approximately 100 billion neurons that generate electrical activity, ha ha ha, because plenty of other tissues in our body generate excitatory neural activity. Skeletal muscles generate action potentials when they contract. Your liver, which is about the same size as your brain, contains a particular type of cell called a hepatocyte that also has an electrical membrane potential. No, what's really exciting about the human brain is what it actually does for us on an individual level. For example, it allows us to acquire knowledge from different forms of perception. It allows us to store some of that knowledge, store some of that information in the form of long-term memories. The brain also controls all of our motor behaviour. You remember motor is a kind of jargon term which means movement. The brain controls even our sort of simplest, most vital elemental movements like respiration and heart rate via the autonomic nervous system. But it also controls all of our complex, more sort of voluntary or willed actions, such as moving the hand to grasp objects. Perhaps, though, the crowning achievement of the human brain is the facility that it has blessed us with to engage in language and communication. Because, we are social animals and we love to express ourselves and communicate. And I guess this is one of the reasons why being locked in a room during the pandemic has caused so much anxiety for so many people. Another important contribution of the human brain is what it does for us collectively, which is it makes us the dominant species on the planet. Because we got big ones, and in this case, bigger is definitely better. So just to give you an indication, these are pictures of the brains of different mammals, all scaled up to the size of a human being. So down here, what I mean by this, down here in the bottom left is a picture of the human brain, and at the top left here is the brain of a rat, if the rat was as big as you and me. And as you can see, the rat's brain is very much smaller than that of the human brain, and indeed of our cousins, the chimpanzees, or of this animal here, the dolphin, all of which happen to be really quite smart animals. Now, I'm not saying rats aren't smart. I mean, rats are an extremely successful species. It's said that when you're in London, you're never further than 10 metres away from a rat. So I'm not quite sure where the rat is in this lecture theatre. I can't see it. I'm alone in this room speaking to you. And rats are actually quite social animals, and they communicate with each other, but their ability to communicate is not as advanced as ours, and certainly their ability to engage in tool use and use technology is not as advanced as ours, because that is sort of one of the crowning achievements of human evolution. The invention of the iPhone, for example. I do have to tell you with some regret, however, that there are some downsides to learning about the human brain. And the downside is that the brain is very, 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 very complicated. It's very complicated indeed. One of the reasons for that is that the brain has many different parts. It's not like the liver in that respect. The liver is just a mass of cells, and those cells are all called hepatocytes or liver cells. And actually they're fantastic cells, they're wonderful. They're incredibly versatile, they do a whole bunch of different jobs. They can detoxify the blood, they can store glucose, they can make hormones, they can make elements of blood clotting agents. They're really, really fantastic cells. They do a whole bunch of different jobs extremely well. But neurons are not like that. Neurons are specialized for one particular function. And for that reason, neurons specialized for a particular function tend to stick together in a particular part of the brain. This is a kind of general concept known as functional specialization. And now, of course, each of those different parts of the brain has a particular name. Sometimes the same part of the brain has several different names, which you often have to know both of in order to remember what we're talking about. And you also need to know what that particular part of the brain is specialized for, what its specialized function is. Also, each specific brain region is connected by axon pathways inside the central nervous system to other brain regions. And guess what? Those axon pathways have different names. And unfortunately, you're going to have to learn what the names of those axon pathways are too. So sorry about that. Look, don't shoot me. I'm only the messenger. But just as an indication, finally, of the complexity of the human brain, it contains approximately 100, in US denomination, billion neurons. 100 billion neurons. A US billion is 1,000 million, by the way. And that is greater than 10 times the number of humans on planet Earth. So that's just an indication of how complicated your brain is. Okay, so finally, let's have a look at a real human brain, which you can see here on the right. This brain has been freshly removed from somebody's skull. And the gross structure of the brain is that it has both a left and a right side, and those sides are sometimes called the hemispheres. And each hemisphere consists of 3 major divisions. The largest of those divisions is this one here. It's called the cerebrum. The smallest is the bit you see sticking out the back, and it's called the brainstem. In fact, this region here would be directly attached to your spinal cord. And the intermediate size division is this one here, called the cerebellum. Now, your cerebrum, which is by far and away the largest part of the human brain, itself consists of 2 major subdivisions. One is called the telencephalon, which means forebrain, and the other is called the diencephalon, which means between brain. And the largest part of the telencephalon is this thing here. It's called the cerebral cortex. And in fact, this is by far and away the largest and most evolved part of the human brain, and actually, in a way, the most interesting, because it's responsible for all the kind of things that we think that the brain does. For example, it's responsible for all of our conscious sensations or perceptions. It's responsible for the control of all of our voluntary movements, and for our various higher kind of cognitive functions, such as memory and language. Another part of the telencephalon, which is quite large, are called the basal ganglia. These are collections of really large nuclei within the base of the cerebrum, which are mainly concerned with the planning and control of voluntary movements. The reason we know this is because people with different types of neurodegenerative disease, like Parkinson's disease or Huntington's chorea, affect different components of the basal ganglia, and these neurodegenerative disorders are characterized by problems with voluntary movement. That's pretty much all I'm going to tell you about the basal ganglia ever. The diencephalon, or between brain, itself has three major divisions to it. The largest of the divisions is called the thalamus, which contains different nuclei. By nuclei, I mean a nucleus is a collection of nerve cells all doing a particular job, which are directly related to, in fact, directly connected via various axon pathways to different parts of the cerebral cortex. Sitting directly below the thalamus is the hypothalamus, because hypo means below, which also contains different nuclei responsible for other jobs, for example, in regulating our various appetites, when to eat, when to stop eating, when to drink, when to stop drinking. And other nuclei within the hypothalamus also regulate our endocrine functions. For example, there are direct connections from some neurons in the hypothalamus to the pituitary gland, which you may recall is the master endocrine organ, and that input regulates the function of the pituitary gland. The final bit of the diencephalon is the smallest. It's actually another gland. Its collective name is the epithalamus, and it consists exclusively of the pineal gland. Now, back in 1600 and something, René Descartes, this is an aside by the way, René Descartes, who was a very famous philosopher, put forward the notion that the pineal gland was the seat of the human soul. One of the reasons he thought that was that the pineal gland sits pretty much at the geometric sort of epicenter of your head. So he thought that's where your soul resides. More recently, we've discovered that it has a rather more prosaic function. It secretes a hormone at night called melatonin, and melatonin passes through your bloodstream and helps to regulate our physiology. And one of the major things it does is help to shut our physiology down so that we can go to sleep. So it has, an important function in controlling what is known as our circadian, that is about-a-day rhythms. Just as a further aside, if you're ever planning to go over to North America in the next few years, I'd strongly recommend that when you get to the USA or Canada, you go to a pharmacy and buy some melatonin tablets, which you can do across the counter in North America. You can't do it in the UK, you have to have a prescription for it. And then when you get on the plane to come back to the UK, that's the really horrible journey, drop a couple of those melatonin tablets as you get on the plane. I promise you, I'm absolutely convinced they really help to deal with the jet lag, to switch your circadian rhythm into the kind of time of UK when you get off the plane the next day. It really sort of takes the edge off that sort of horrible feeling of jet lag that you get travelling from west to east. I always drop melatonin tablets when I come back from the USA or Canada. Okay, so what is the brain made of? Well, I think you may be familiar with this kind of general term, grey matter. By definition, grey matter contains the cell bodies and their dendrites of neurons, along with the synaptic inputs they receive from the terminal axons of other neurons. And hence the grey matter is a site of information processing within the brain. The way in which the neuron cell bodies are laid down differs from one part of the brain to another. So, for example, in the cerebral cortex, which I've already mentioned is the big bit of the cerebrum, the neuron cell bodies are laid down in sheets or very obvious layers, whereas elsewhere in the brain they're just sort of clustered together in groups, known as nuclei in the plural or a nucleus in the singular. And that's the arrangement that you see both in the basal ganglia and in the thalamus. The other major component of the brain is number 2, the white matter. Again, a term with which you may be familiar. And by definition, the white matter contains the axons of neurons, which are also known as nerve fibers, and their associated myelin sheaths that are formed by oligodendrocytes. The reason the white matter is white is because the myelin sheath is just basically the cell membrane of the oligodendrocytes wrapped repeatedly around the segments of central nervous system axons. And the membrane of a cell, including oligos, is just a lipid, so it's fat. And that's why the white matter is white. It's mainly fat. Now, the white matter inside the brain is mainly a whole series of routes for direct long-range connections or projections between neurons in different grey matter regions. And therefore, the major function of the white matter in the brain is for information transfer. And I'm just going to mention a couple of really huge white matter pathways which are characteristic of the human brain. One is called the corpus callosum. This is a really huge bundle of axons. In fact, over 200 million axons that directly connect the left side of your cerebral cortex to your right cerebral cortex in each of the cortical hemispheres, so the two sides of the brain know what each is up to. Another really huge white matter pathway is this one here. It's called the internal capsule, again with over 200 million axons in it, which directly link neuron cell bodies in different nuclei of the thalamus to the different parts of the cerebral cortex. This next picture is a cartoon, or a kind of schematic, that I put together a few years ago when I had a couple of hours to spare. I don't know how the hell that ever happened, which I hope may be of help in getting this distinction between the grey and white matter, sort of a little bit clearer. So here in this ellipse are a series of these yellow things. They're all looking really spiky. That's because they are the cell bodies of different neurons, and the spiky bits are their dendrites. You see? Got it? The orange blob, by the way, is the nucleus inside the cell body containing the cell's DNA. And as you see, all of these cells are exactly the same size and they're all the same shape, and that is because they're all doing the same job. They're functionally specialized for a particular role. So those cell bodies and their dendrites are forming the grey matter, or nucleus in this case. And the yellow line coming out of each of these cell bodies is therefore their axons, which are sending impulses down their axons to target cells somewhere else. And those axons therefore are running within the white matter. They will have myelin sheaths around them. I don't know if that helped at all. This is a horizontal section. It's from a post-mortem of a human brain cut in what is known as the transverse or coronal plane, with the left side of the brain here and the right side of the brain over here. And this post-mortem slice has been stained slightly to accentuate the difference between the grey and white matter. So you can see on the outside of the brain here, for example, all this curly stuff. That's the cerebral cortex, with grey matter here on the outside. Here, deeper in the cerebrum, is a big chunk of grey matter here and here. These are two large nuclei of the basal ganglia. They're known as the caudate and putamen nuclei, but you actually don't really need to know that. And then you can see the white matter. Here is the white matter in the inside of the cerebral cortex. This thing here running across from left to right or right to left, that's just a small part of the corpus callosum, the interhemispheric pathway that takes information from left to right cortex and from right to left. And down here is a little hint of part of the internal capsule, the white matter pathway taking information from thalamus to cerebral cortex. Okay, so that was a sort of general introduction to the brain. I'm just going to finish off a little bit here on the brain by telling you just a little bit more about the cerebral cortex as a prelude to a later lecture, which is going to be about an hour on this particular topic. Right. We actually saw before in that previous slide that the cerebral cortex in humans is deeply folded. It's a very convoluted structure with ridges and furrows within it. And one of the reasons that the cerebral cortex in humans is so deeply folded and convoluted is to try to pack as much of this material into our skulls as is possible. If you could take an iron and sort of flatten out the cerebral cortex, it would have an average surface area of about 1,250 square centimetres, which is about the surface area of a small pillow. There are a lot of words on the screen, but here is a photograph now of a human brain. We're looking at the left side of the brain here. The front is over here, and the back is over here. Most of this curly stuff that you can see here, this is all cerebral cortex, this folded or convoluted material that we're currently talking about. And for the record, this is the brainstem sticking out the back, and here is the cerebellum. Now, the bumps on the surface of the cerebral cortex, or the ridges, are known as a gyrus in the singular, or as gyri in the plural. And in between the ridges are quite deep furrows, or slits, which are known as a sulcus in the singular, or sulci in the plural. And in a way, this is kind of like a mountain range. You know, the hills or the tips or the ridges of the mountains are here, the gyri, and then the valleys in between the mountain range are the sulci. And your cerebral cortex is divided into 4 major lobes. It isn't really obvious from this picture that there are 4 separate regions to this tissue, but now in the next picture I'm going to explain how this comes about. Here are the words, and now I'm going to tell you what those words mean. So this is another view of the left side of the human brain. In this case, though, the cerebral cortex has been and its 4 lobes have been rendered in different colours, and we can see the brainstem and the cerebellum again sticking out the back. Okay, so there's one sulcus that we can see here. It's found about halfway from the very front to the very back of the cerebral cortex, and for that reason it's called the central sulcus. This furrow in the side of your brain is extremely deep. If we had a real human brain in front of us, you could take your index finger and poke it all the way down the central sulcus before you reach the bottom. And the important thing about the central sulcus is it's an important landmark. It divides the lobe of the cerebral cortex here in blue in front of it, which is known as the frontal lobe, from the lobe of the cerebral cortex behind it in green, which is the parietal lobe. The main function of the frontal lobe is to control our voluntary movements, so that's what it's functionally specialised for, whereas the main function of the parietal lobe is in somatic sensation. Remember, somatic sensation involved things like touch or tactile sensations, thermal sensations, pain, and joint sense, knowing the position of your joints in 3-dimensional space, otherwise known as proprioception. Those are the somatic sensations. And the parietal lobe is responsible for conscious perception of those types of sensation. Also in the lateral surface of the cerebral cortex is another really deep sulcus that you can get your index finger all the way down. And quite conveniently, as indicated here, it's called the lateral sulcus. It divides the frontal and parietal lobes above from the orange temporal lobe below. Most of the temporal lobe, at least that you can see here, is predominantly, these bits up here, mainly concerned with hearing or auditory functions. And the 4th and final lobe is the one which is actually most important to you as future optometrists. It's the one shown here in red, and it's called the occipital lobe. on this lateral surface of the cortex, there's no sulcus that demarcates where the occipital lobe ends and the parietal lobe begins, or where the occipital lobe ends and the temporal lobe begins. But anyway, the important thing about the occipital lobe is that it's mainly concerned with vision. And indeed, you need an intact occipital lobe in order to be able to see, to have any form of visual perception. Right. Okay, so that was kind of the point that I wanted to leave those bits of discussion of the human brain at. I'm going to take a little brief pause, mainly to take a sip of water, and also to give you a moment to kind of relax. And then we'll talk a little bit more about, I'm going to change gear a bit and talk about some methods for studying the human brain. In fact, there's going to be more of this later on in the module, in fact in term 1 and also in term 2 as well. But again, by way of a kind of general overview before we get to some more detailed stuff later on. So just a brief pause then, and then I'll be back. Okay, so I'm back. And I'm going to continue by just telling you a little bit about some modern methods for imaging the structure of the living human brain. One of the reasons that we tell you about this kind of thing on this module is because the GOC think that you ought to know about this kind of stuff. Do you know who the GOC are? Yep, the General Optical Council. Well, of course, they're fully aware that you're never going to actually use these particular techniques to visualize the living brain of a human being, but they do feel that you ought to know a little bit about their existence and how they work, because sometimes you might get the opportunity to see one of these images and be asked to interpret it, and as part of a diagnosis of somebody. And we'll look at a couple of clinical examples in the next 10 minutes or so, where techniques for imaging the brain are of use for people practicing optometry. Okay, so there are two sort of accepted and basic methods for visualizing the structure of the living brain. They're called X-ray computed tomography, or CT scanning, and magnetic resonance imaging, or MRI scanning. The principle behind the X-ray CT is that a person lies in a scanner and an X-ray beam is fired repeatedly into their head, but at different angles. So the X-ray beam is actually on a kind of circular gantry that rotates around the person's head, and it fires an X-ray for each of its particular positions, and the X-ray passes through the person's head with different levels of attenuation. As you're probably aware, there are some hard tissues inside our bodies, an example of which is bone, and another is tooth enamel, which attenuates an X-ray beam. In other words, it doesn't allow the beam to pass through to the kind of photographic plate on the other side. And by convention, in X-rays, those high attenuation structures like bone and tooth enamel appear as white in X-rays. However, much softer tissues, I'm thinking of things like air and water inside the head, allow the X-ray beam to pass through completely unattenuated, so very low attenuation. And those appear in kind of black in conventional X-ray images. And the same sort of thing kind of applies to CT scans as well. Although some of the intermediate soft tissues in the head, of which the brain is an example, kind of come up on a little bit of a grayscale. And one of the really useful things about the CT scanning is that each of those repeated X-ray beams produces its own single image of the head, and the computer bit sticks each of those images together, those images in the form of kind of slices through the head, and the word tomos means slice, in order to produce a kind of 3D reconstruction. So you get these kind of slightly better, actually much better than X-ray views of a person's head, but also in a series of slices that you can look at in 3D. This particular approach is particularly useful for looking at really kind of gross pathologies, things like strokes or tumours inside somebody's head, if those are suspected in the patient that's being examined. The second technique, which is MRI, is maps hydrogen nuclei and water concentrations inside people's heads. Again, the person lies in a scanner. I don't know if any of you have been in an MRI scanner before. I've done it several times as part of a kind of a research project. And the scanner passes, again in different directions, a really powerful magnetic force through the head, because the magnetic field around the MRI scanner is several thousand times greater than that of the magnetic field of the Earth. And it causes hydrogen nuclei to kind of spin in the same direction as the direction of the magnetic pulse. The MRI also uses computerized systems to compile sequential slices of the head and also to form 3D images. But the real advantage of MRI over CT scanning is that MRI produces much differentiation, better differentiation for softer tissues. In fact, you can even see differences between the grey and the white matters in people's brains. So this is a technique which is more useful for looking at rather more subtle pathologies of the brain. So these two pictures on the left and right are both MRI scans through, as far as I know, a normal person's head. This one over here on the left, this is a kind of horizontal section, and so is this. And so this is the front part of the head, and this is the back or posterior part again. This is the front part, and this is the posterior part. Now, the radio frequency pulses that are applied from the magnetic field around the head cause the odd proton number, i.e. in hydrogen or water, to temporarily align with the external source, or to resonate, hence resonance, with particular time constants is T. In time constant 1, all of the protons are aligned with the direction of the magnetic pulse. And in this particular version, T1, the white matter inside the brain appears lighter than the grey matter surrounding it. So that's kind of the normal arrangement, where the white matter is white and the grey matter is grey. And in the second constant, which is called T2, the alignment of those protons with the direction of the beam becomes completely lost. And under those circumstances, the white matter appears darker than the grey matter around it. In other words, the kind of contrast or polarity is reversed. And those two different types of time constant, either T1 or T2, are used to look at pathologies depending on what is suspected may be wrong with the individual concerned. So how do we know about the different functions of different brain regions? Things that I've been telling you about so far in the context of functional specialization. Well, there have been two sort of major approaches that have revealed how it is that different brain regions are involved in different functions. One is a really ancient one. It's clinical neurology, examining patients who are still alive but have some form of brain damage, as a consequence of which they've now lost the ability to do something or to sense something that they could do before. So examining surviving patients who have had problems like a stroke, where an artery has become blocked or has burst, some form of brain trauma, a direct brain injury or forms of disease, which result in selective deficits in neural function, which are caused, by the localized region of brain damage. Brain damage is collectively known as a lesion. The other approach is a much more modern one, and it's functional brain imaging. And it's a kind of complementary approach, but using healthy, neurologically intact volunteers, actually quite often medical students. And in this approach, functional brain imaging, again, people are put in scanners, and what the scanner is looking for is localized increases in brain activity while they're performing a specific task in the brain scanner. And those localized increases in brain or neural activity are associated with an increase in oxygen utilization in the particular area. So actually what the scanners are looking at is parts of the brain that are using more oxygen, actually sometimes more glucose, because that's all that neurons eat. Their diet's very restricted. Neurons eat either oxygen or glucose. And what the scanners are looking for are regions of the brain in which the oxygen consumption or glucose consumption has gone up, which is indicative of that part of the brain is neurally active. It's a kind of substitute or proxy for actual neural activity. So here are a couple of pictures of individuals who have got brain lesions. So this is taken from clinical neurology, and in these two particular cases, the patients have undergone an MRI scan, a structural brain scan, and these are T2 weighted images taken from those two patients, where you can see here, for example, the white matter is quite dark, and the grey matter, again, this is cerebral cortex, is lighter. This person on the left here, as you can see, has got a major problem back here, back in the occipital lobe, on the right side of their cerebral cortex. In fact, the problem that they've had is a major artery has hemorrhaged or burst, and that's what this black stuff is. It's all kind of congealed blood, and also some of this material here too, in their right occipital lobe. So if their right occipital lobe has had this stroke, what it means is that the neurons in the occipital lobe have died, they're no longer working, and as a consequence of that, this person is going to have a major problem. And because it's the occipital lobe, they're going to have a serious loss of vision. In fact, this stroke is so large that they didn't really need to do an MRI scan. This would have been revealed quite adequately with a CT scan. So I'm not sure why they did MRI. In general terms, CT scans are cheaper and they're easier to do than an MRI. The brain image on the right, however, again a horizontal slice through another patient's head, this is the front part of their brain and this is the back, contains a subtle pathology that you wouldn't be able to see in a CT scan. And that pathology is those little blobs of white that you can see, and there are lots of them. There are multiple blobs of white here, 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 all within the white matter of this person's brain underneath their cerebral cortex. Actually, that's why they've used a T2 weighted scan here to show up the intact white matter in black so that they can see these plaques, these sclerotic plaques of damaged white matter very clearly in the MRI scan. Because unfortunately this person is suffering from multiple sclerosis. Each of those white dots are regions of demyelination occurring in the white matter linking the thalamus to the cerebral cortex. And there are lots of them. So this person is going to have multiple neurological deficits. I mean, I can't tell you what they are because I don't know exactly which axon pathways have been affected. But this is going to be this person is going to be suffering from multiple neurological deficits as a consequence of their sclerosis. Really, really nasty disorder that I hope none of you have any experience of ever, both for you and or members of your family. Okay, so what we've been looking at in those last few images was how to look at the brain structurally, but there are also modern techniques for how to look at the brain from a more functional level, again, either in healthy, intact subjects or in people suspected of having some sort of neurological disorder. And the two main approaches for looking at the function of the brain are positron emission tomography, or PET, and functional magnetic resonance imaging, or fMRI. The principle behind these two techniques, again, is to look at oxygen or utilization within different parts of the brain, with high levels of oxygen utilization, implying an area of high brain activity, as I mentioned before, as a kind of proxy for that. Positron emission tomography involves injecting a radio-labeled isotope, in fact it's the 15 version of oxygen, into usually an artery of the patient. And when it reaches a region of the brain which is using a lot of oxygen, the positrons annihilate on collision with electrons, producing two gamma rays which travel in opposite directions to detectors which are in the scanner around a person's head. So this is a method for localizing regional changes in cerebral blood flow, or RCBF. In fact, positron emission tomography is only used sparingly because you have to inject this radio-labeled isotope into a person in order to perform the technique. And in general terms, clinicians don't like to inject radio-labeled isotopes into people. So by far and away the most common and technique for looking at the functional brain is fMRI. Again, this is a non-invasive technique where you put people in an MRI scanner, but you look at different resonances produced by hemoglobin versus deoxygenated hemoglobin that produce a signal that enables the person to identify or localize changes in blood oxygenation level dependent or bold signals, regions where the brain is using lots of oxygen. Just to give you an indication of how these functional approaches work, and this is just kind of an illustration. Say you wanted to find out what part of the human cerebral cortex was particularly concerned with visual function. Okay? The general approach would be to put a healthy, neurologically intact person into a scanner, and in the test situation here, they would have their eyes open, and they'd probably be looking at something inside the scanner, so they'd be using their visual system. And then the scanner would record the brain activity or the neural activity throughout their whole brain, based on regional cerebral blood flow, or BOLD if it was fMRI, when their eyes were open and they were using their visual system. So they'd make that scan. Then in the next scan, the subject would close their eyes, so they wouldn't be using their visual system at all. And then another brain scan would be taken, looking at all brain activity, with the eyes closed and the visual system not working. And then the brain scan taken in the test situation, the control situation would be subtracted from the test, and that would, by subtraction, leave the particular regions of the brain, or cerebral cortex, that were specifically activated by the stimulus, in this case by the visual stimulus. Okay? So eyes closed, brain activity subtracted from eyes open brain activity gives you the regions of the brain that are active when you're seeing and you're using the visual parts of your cortex. And in this series of images, that's exactly what's happened. It's showing the increase in regional cerebral blood flow, particularly in the occipital cortex of a person who was lying in a scanner, first with their eyes closed, then minus the activity shown with their eyes open, then minus the activity with their eyes closed. Again, these are horizontal slices through different levels of the cerebral cortex. This is the top, and we're working gradually down here towards the bottom. The regions that you see here in this image, or in these images, which are really dark red with white, are regions of increased brain activity associated with the eyes open versus eyes closed condition. And I've put white arrows here to indicate where they are. As you see, they're back here, at the very back end of the cerebral cortex, specifically within the occipital lobes. In other words, this is a demonstration that the occipital lobes are particularly active during visual perception. Now, actually, the main reason I showed you these images is that the brain in them is mine. I thought you'd be happy to know that I have one, and that at least a few years ago, the visual parts of it seemed to be working quite well. Okay, so we'll just finish with a sample multiple choice question, the type of thing that we're likely to ask you in the exam that is due to take place hopefully in May or June of next year. It's a really simple question, just to kick us off. Which of these cerebral cortical lobes is mainly concerned with initiating voluntary movements? They're in alphabetical order: frontal, occipital, parietal, temporal. I'm not going to give you the answer. I'm pretty sure that you probably know it already, but please don't say B, occipital, because that's wrong. Occipital, of course, is vision. Okay, so that's lecture 1. We will now take a brief pause, and then the echo will start up again with lecture number 2, in which we will look a little bit more at the human brain. Okay, cheers. Okay, so this next lecture continues a kind of general overview of the human brain, but it's mainly concerned with how to keep it alive and well. Now, the first important point to make is that the brain is a very delicate and a very hungry organ. It's very delicate and therefore needs different forms of protection. And it's a very hungry organ, as kind of indicated by the fact that all three parts or three major divisions of the brain shown in this picture are quite obviously really quite pink, aren't they? There's a lot of pink around here. And the reason for that is that all parts of your brain are absolutely stuffed full of capillaries. And this is an indication of the fact that it's a hungry organ. So, for example, just to sort of put this into context, your brain on average weighs something between like 1.5 and 1.7 kilograms, which, depending on your overall weight, is something like 2% of your total body weight. But it uses about 20% of our metabolic resources. So, you know, a tenfold or an order of magnitude more of our metabolic resources go to keeping our brains alive and well than based simply upon its size. So the aims of this lecture are to look at the protective mechanisms for keeping the brain healthy. So we'll look very briefly at the bones of the skull that are particularly responsible for protecting your brain. We'll look at some membranes that are found on the outside of the brain tissue. These are also protective membranes. They are known as, there are three of the membranes, and they're known as the meninges or maters, the word mater meaning mother. We'll look at the blood supplies to the brain and a little bit about the venous drainage. And finally, we'll look at the fact that your brain contains really quite large spaces within it. Those spaces or cavities are filled with a fluid which is known as cerebrospinal fluid or CSF, and the spaces are known collectively as the ventricular system. So we'll look at those at the end of the lecture. So first of all, let's look at how your brain is protected, particularly by bones of the skull. Now, in fact, your skull comprises 22 separate bones. 6 of them are singles. There are only 6 of these single bones, and they tend to be at the midline of your skull, and then 8 of them are paired, so you've got one bone on the left and a counterpart bone on the right. 8 pairs is 16 plus single, 6 is 22 in total. And in fact, your skull comprises 2 main regions. There's the roundy bit on the top, which is known as the cranium or cranial vault. And it's the cranium or the vault that actually covers the brain. And the other part of the skull is mainly the bones of the face, and they're called the facial bones. The cranial vault, which comprises 8 separate bones, has these 4 bones which are single, they're unpaired. There's a frontal bone at the front, an occipital bone at the back, and these 2 bones, known as the sphenoid and the ethmoid, which are at the front of your skull, and in fact form parts of your orbital cavities. In fact, the back of the orbit and the medial wall of the orbit. Back of the orbit is sphenoid bone, medial wall of the orbit is ethmoid, but also protect part of your brain. The other 2 bones are paired, so you have one of these on the left and one of these on the right, and they're called the parietal bones and the temporal bones. So those are the bones that are actually completely encasing your brain. The other bones that are shown here on the slide are concerned with more with the face. I mean, just to give you an indication of what some of them are, the mandible is the name of the lower jawbone. The maxilla is the name of the upper jawbone. And the ones that you see here with asterisk by them are all bones that contribute to the formation of your eye socket or orbital cavity. Unfortunately, there isn't a single orbital bone that holds your eye in the eye socket. In fact, there are 7 separate bones that contribute to the orbital cavity. You know, if you haven't already been told what they are, you'll find out in some other module, perhaps in eye disease or something. But these are the bones, the 7, frontal, sphenoid, ethmoid, contribute to the orbital cavity. So does the maxilla, the zygomatic, the lacrimal, and the palatine. Okay, so this is a version of a picture that I showed you in the previous lecture. It's just a reminder that the cerebral cortex comprises 4 main lobes. We've got the frontal lobe here in blue, the parietal lobe here in green, the temporal lobe here in orange, and the occipital lobe here at the back in red. And the bones of the skull that form the roundy part of the cranial vault have exactly the same names. They're called the frontal bone, the parietal bone, the temporal bone, and the occipital bone. And they pretty much directly overlie those 4 different lobes of the cerebral cortex. So those are 4 of the bones protecting your skull, your brain: frontal, parietal, temporal, and occipital. And here at the midline, just behind and helping to form part of your orbital cavity, here's the ethmoid bone. It's the bone which actually forms the midline, most of the midline of your eye socket. And here is the sphenoid bone, here in yellow on the side, sticking out just here, part of which also forms the apex, or the very back of your eye socket. So those are the 8 bones. Here are the singles: frontal, occipital, ethmoid, sphenoid, and here are the 2 paired ones, because there's a temporal bone on the other side and a parietal bone on the other side, protecting the brain. If you look on the inside of your skull, we're looking here at the floor of the skull, 5 of those bones, all of them in fact except for the parietal bone, also contribute to the ventral part of your skull, the underside of your skull. So here's the frontal bone at the front, here's the occipital bone at the back, here are the 2 temporals, that's the location of the ethmoid here, and here is the sphenoid bone situated right here. Your orbital cavity would be underneath where I'm pointing these arrows. Okay? So your orbit is just underneath the frontal bone here. On the other side of the picture here on the left, you can see the underside, that is known as the inferior or ventral surface of the brain. And I've shown it in this picture just to indicate just how sort of complicated it is. There's lots of kind of bumps and nooks and crannies in the underside of your brain tissue. But the point is, if you take that brain and flip it over, turn it upside down and stick it into the front, into the cranial cavity here, all these bits of the brain fit really perfectly into these nooks and crannies in the floor of your skull. Because the floor of your skull has 3 major pairs of spaces within it. There's a big pair of spaces here, which are known as the anterior fossas, a big pair of spaces here at the back, which are known as the posterior fossas, and 2 big spaces here and here between the front and back, which are known as the middle fossas. The word fossa just means space. So you've got these 2 fossas frontal, 2 fossas posterior, and 2 fossas medial. If you take the brain and flip it in, you'll see that the frontal lobes fit really perfectly into these spaces here in the frontal fossas. You remember these things here, these little blobs? Those were the olfactory bulbs that receive input from the first cranial nerve, the olfactory nerve. And we did this last year in the cranial nerves lecture and lab. Those olfactory bulbs fit really perfectly into these little grooves here, formed by the ethmoid bone, and those grooves were known as the cribriform plate. Do you remember that from last year, cribriform plate? If you flip the brain over, the occipital lobes, and indeed, which are underneath here, and these 2 things here, which are the cerebellum, they fit perfectly into the posterior cranial fossas. And if you flip the brain over, the temporal lobes here and here, they fit perfectly into the middle cranial fossas. And indeed, the pituitary gland, which is just situated here, we did the pituitary gland again last year in first year. If you flip the brain over, it sits perfectly in a little bony well here, which is part of the sphenoid bone. That is called the pituitary fossa, or hypophyseal fossa, and is a special well designed specifically to house your pituitary gland. In fact, the shape of the underside of your skull, the shape of these bones, is formed by the shape of your brain, not the other way round. And so those bones on the underside are also therefore protecting your brain from below. Okay, so I mentioned before that the brain is very delicate. I've also told you, haven't I, that the brain is bilaterally symmetrical. It has a kind of left and right hemispheres which look pretty much from an anatomical point of view as if they're the same. But notice in this picture of the brain that's been freshly removed from this person's skull, it seems that the cerebellum here is mainly found on the right side of the brain with very little cerebellum on the left. But actually that's just an artifact of the delicacy of the brain. It's just that the cerebellum has collapsed over here towards the right because the brain is a very delicate tissue. In fact, it has kind of the consistency of that set yogurt, fromage frais. I mean, if you had a teaspoon, you could dip it into the brain here and just sort of spoon bits out of it, because that's the sort of nature of its consistency. I'm not suggesting for a minute that you want to do that, but you could if you were desperate. So, apart from the skull bones, the brain also has this covering of meninges, of membranes around the outside to hold it into place. And this, on the left, shows the thickest of the layers of the meninges, which is called the dura mater. The word dura means tough. So this is the tough mother here. This is a person's head. The vault of their skull has been removed, and this material here that you see is the really tough membrane, the dura mater, which has its own large blood vessels in it that are completely surrounding the outside of the brain. The brain is underneath the dura. Okay, so these three membranes are known collectively as the meninges and also as the maters. The outer layer of the membrane is the one I just showed you. It's called the dura mater. The inner is called the pia mater, and the middle layer is called the arachnoid mater. The outer dura mater is the really tough mother. It's made of a very tough, fibrous, collagenous material, in fact with the collagen fibres laid down in separate layers. And the dura mater is actually fused to the connective tissue on the inside of your skull bones. As a consequence of that, the dura fuses your brain to the inside of your skull and prevents you from moving, prevents the brain from moving on your head when you're moving your head around as part of the protective mechanism. Because if your brain were to move inside your skull when you move your head around, it would slam into the skull bones and cause bruising. So one of the major functions of the dura mater is to suspend your brain inside the skull, which also helps to reduce its overall weight and its movement. And it also extends double folds of this dura into quite large spaces or fissures on the outside of your brain. One of those fissures is called the longitudinal fissure. It's the slit that runs between the left and right cerebral cortices, this one here. So there's a space between the left and right cortex that runs kind of in a longitudinal direction, like this, and it is called the longitudinal fissure, and the dura mater dips down into that fissure to help suspend your brain within the skull. That double dip of dura mater has a name. It's called the falx cerebri. Falx means sickle, and cerebri means brain. So it's a sickle-shaped piece of dura that runs down the longitudinal fissure and helps to hold your brain in position. Another one is called the tentorium cerebelli. That's a big double sheet of dura that you find between the occipital lobes of your cortex and the underlying cerebellum. And as we'll see, these large double folds of dura also contain, enclose and protect really large veins that you find within these fissures on the outside of the brain, but that's to come. The pia mater is the innermost layer. It's a very, very fine and delicate membrane that closely invests the entire brain surface. In fact, it follows all the various convolutions and contours of the brain surface. it's so delicate that you'd need a pair of fine forceps to get hold of it. But it contributes in some ways to the blood-brain barrier. The third, or in fact middle layer, is called the arachnoid mater. Arachnoid means spiders, so this is the spidery mother. This is also quite a delicate membrane. It's thinner than the dura mater, but thicker than the pia, and it wraps itself over the surface of the brain a little bit like cling film, creating in some places a space between it, a very obvious space between it and the brain surface. And that space is known as the subarachnoid space because it's underneath the arachnoid and next to the pia. And in your subarachnoid space, there is cerebrospinal fluid, or CSF, and also it's a space where the larger blood vessels, arteries and veins are found on the surface of the brain. So here they are. This is a scanning electron micrograph of the three layers that form your meninges. This is the dura mater on the outside. You can see it's quite thick. It's actually about 0.5 millimeter in thickness. It's comprised mainly of separate sheets, layers of collagen and elastic fibers. This, by the way, is the brain underneath. This is a scanning electron micrograph. That's a neuron cell body. So this is brain tissue. And running along the surface of the brain tissue is this really, really fine membrane here. That's the pia mater. And here is the middle layer, the arachnoid mater, the spidery mother. And here, between the arachnoid and the pia, is the subarachnoid space, which on the outside of your brain contains CSF, cerebrospinal fluid, and blood vessels, as indicated by this thing here. So that's what's in your subarachnoid space. You can see in this picture there also appears to be a space between the arachnoid and the dura, but that's an artifact of the tissue processing. There is no space between your arachnoid and your dura mater. The two membranes are sealed next to each other. Although, unfortunately, that space can appear in people who have head injuries that have caused the blood vessels in the dura to rupture. So, for example, you see these blood vessels here that are running actually in the dura mater. If you have a head injury that causes those blood vessels to rupture, the blood will leak into the potential space between dura and arachnoid, and that is called a subdural hematoma. And a subdural hematoma tends to expand very gradually over a period of hours, so that people who have had a head injury will quite often get up, and not even lose consciousness, but then over a period of time, the blood will build up in the subdural hematoma, push against the brain substance, and then they will lose consciousness. And quite often, this can unfortunately be a cause of death. Natasha Richardson, an actress who was some years ago married to Liam Neeson, a famous massive actor who's always in these violent films like Taken 1, Taken 2, Taken 3. Unfortunately, she had an accident in which she fell, banged her head. She got up and walked away, but a few hours later, she was in hospital in a coma, and unfortunately died of a subdural hematoma. That was an aside, by the way, an unfortunate one. Okay, so those are the things that I wanted to tell you about for protecting the brain, the skull bones and the meninges. Now we're going to move on and talk about where the brain gets its blood supplies from. And there are two sets of paired arteries that are responsible for supplying the brain with blood. Two of those paired arteries are the internal carotid arteries, and the other pair is the vertebral arteries. The internal carotid arteries mainly supply the front half or anterior half of your brain. They enter the skull through a couple of holes, because there's the left and right carotid, called the carotid canal, and when they get inside your skull, they form three major branches, each of these arteries. Okay? So the first intracranial branch of the internal carotids is known as the ophthalmic artery. And as you can probably guess, this is a really important artery for optometry, because this is the artery that enters the orbital cavity at the back through the optic canal, and the ophthalmic artery splits into a whole bunch of branches of its own and supplies pretty much all of the tissues in your eye, including the retina and all of the contents of your orbit. The other two major branches of the carotid arteries supply parts of your cerebral cortex, and they're known as the middle and anterior cerebral arteries for that reason. The vertebral arteries also enter your skull. In fact, they enter the skull at the base of the skull through the big hole, which you may recall is called the foramen magnum, and their job is to supply pretty much the posterior or back half of your brain. Actually, the vertebrals also have the branches that go down and supply your spinal cord as well. When they get into the skull through the foramen magnum to supply the brain, the two vertebrals come together to form a single, really stonking artery at the very base of your brainstem, and that's called the basilar artery. And this artery has a bunch of different branches, actually, that mainly supply your brainstem and your cerebellum, but also gives rise to a terminal branch, which is called the posterior cerebral artery, which mainly supplies the back end of your cerebral cortex, particularly your occipital lobe. So the posterior cerebral artery is the blood supply to your occipital lobe, which of course is the bit involved in vision, and therefore is a really, really important artery. This is a schematic. We're looking here at the right side of somebody's brain or head. This over here is the front and this is the back. And this is just an indication of the carotid arteries running up through your neck, where they are in the form of the common carotid artery. The common carotid splits in your neck to form an external carotid, which mainly supplies blood to your face. And here's the internal carotid running up through the neck, entering through the carotid canals, doing this kind of funny little S-bend thing. Just here, this little loop. Actually, that loop is found within a venous sinus, which you're going to hear more about in the future. So I'm just going to mention it now. It's called the cavernous sinus. And it also gives rise, here you see, to the ophthalmic artery that enters the orbit to supply its contents. And here is the middle cerebral artery, one of its terminal branches, running up and fanning out over the surface here, the lateral surface of the cortex. And the anterior cerebral actually runs forward and supplies most of the medial surface of the cortex, a bit you can't see because it's on the other side of the screen. This is the vertebral artery entering the skull here through the foramen magnum. There's a branch going down into the spinal cord, but here it's forming the basilar artery underneath, and here is an indication of a terminal branch going back to supply the posterior part of your cerebral cortex. The cerebral artery territories, the bits of the cerebral cortex that are supplied by these three cerebral arteries. I just thought, just in brief here, we'd kind of do a sort of a so what. I mean, who cares about what part of the cortex the anterior, middle, or posterior cerebral arteries supply? So I just thought we'd have a quick look. This is the left cerebral cortex that we're looking at, but this is the medial surface, the inside bit that we've not seen before. So this is the front anterior, and this is the back posterior. And most of all of this anterior part of the cortex is supplied by the anterior cerebral artery, and this bit back here, this would be the occipital lobe, is supplied by the posterior cerebral artery. Here we're looking at the right cerebral cortex. Again, this is the front, this is the back, but now the lateral surface. So this is the region supplied by the anterior cerebral, this is the region supplied by the posterior, and this kind of greeny colour is the region supplied by the middle cerebral artery. So one of the things that we could say is, well, okay, what would happen if you had a blockage or a rupture, say, of the left posterior cerebral artery? In other words, here's the origin of this artery coming up, and this artery is now blocked, so there's no, or it's ruptured, so there's no blood supply to the neurons here in the occipital lobe. Would that have a serious consequence? You bet. all the neurons involved in visual perception would be dead, and you'd have a visual field loss. In fact, you wouldn't be able to see anything in the right half of your field of view. Let's talk about the middle cerebral, if it was the left middle cerebral artery that supplies this kind of tissue. So bits of the frontal lobe, bits of the parietal lobe, bits of the temporal lobe. Remember, the frontal lobe's involved in voluntary movement, the parietal lobe in somatic sensation, temporal lobe in hearing. So yes, there would be losses of all of those functions as a result of a blockage or rupture of the middle cerebral artery. So it becomes important to know what the arterial territories are for that particular sort of clinical reason. And it's something that we'll explore in more detail in future lectures. Now, those two arterial supplies, the internal carotid and vertebral basilar, are actually joined together by three communicating arteries. One is called the anterior, and then there are two posterior communicating arteries. So they're actually they're not completely separate. They're joined, and those to form a circle of arteries that you find at the very base of your brain, which is known as the circle of Willis. And the general idea behind this is that these communicating arteries operate to form a kind of shunt. So I'm going to skip the words here and show you a couple of pictures and try to talk you through it. So here on the right, we've got a photograph of a real brain with the various arteries comprising the blood supplies to the brain and the circle of Willis shown. So this is the frontal lobes of the cortex. These are the temporal lobes. That's the cerebellum. And here are the arteries. Look, you can see two vertebrals coming in, joining together at the base of the brainstem. This is part of the brainstem, to form this single, really large basilar artery here at the base of the brain. And here are a couple of branches going off here and here to supply parts of the cerebellum. And here is its terminal branch, here on the right and here on the left. That is the posterior cerebral arteries, as indicated also over here in this kind of schematic. Two vertebrals, single basilar, and here is the posterior cerebrals going back to supply your occipital lobe. This open circle here and here, these are cross-sections through your internal carotids. Here you can see a branch running anteriorly, so that's an anterior cerebral artery. And on this side you can see this branch running out here laterally. That's the middle cerebral artery from this side. So you've got the middle cerebral here and the anterior cerebral here. So these are the parts, and again you can see them over here, middle cerebral, anterior cerebral, and internal carotids for the bits that supply the anterior parts of your brain. But here you see is a small artery that bridges between the two anterior cerebrals. Just this single one, that is the anterior communicating. So what this means is that blood can be shunted from here, the right anterior cerebral artery, here into the left anterior cerebral artery. And that might be useful if there was some sort of problem with the blood flow in one of those arteries. Say, the blood flow running through this right anterior cerebral was reduced for some reason. Maybe the person's a bit old and they've got atherosclerosis and the blood isn't flowing as quickly as it should do. Then blood in this unaffected left artery could shunt blood into the right anterior cerebral and compensate for that loss of blood flow. And that's really important because, the neurons in your brain need to be constantly perfused with blood in order for them to fulfill their functions. If your blood supply is reduced or indeed absent for longer than a few minutes, those neurons will start to die. And they won't regenerate, they won't recover, and therefore there will be permanent brain damage. The posterior cerebral arteries, of which there are a pair, are thought to act as a shunt between the posterior and anterior supplies. So, for example, you can see here, this is the posterior cerebral artery, here on the left. That narrower artery there is the posterior communicating, and what it's doing is to communicate directly here with the internal carotid artery, here on the left, and therefore is able to shunt blood from the posterior cerebral into the internal carotid supply, or in the opposite direction if needed. So again, if there was an insufficiency of blood flow through this internal carotid supply, you can shunt blood through the posterior supply to help compensate for that. Just to put this into context, I just want to tell you, again, this is something that you don't actually need to know, but it just shows how important the shunt might be. You may be aware that around the world there are various research laboratories that are trying to develop drugs that can be given to patients, to human patients, shortly after they've had a stroke, either due to a blockage or to a rupture of an artery supplying a different part of their brain. The reason for giving these particular drugs is to try to keep the neurons alive for a period of time, and healthy despite the loss of blood flow. And so these research laboratories use animal models in order to investigate the efficacy or power of these particular drugs. So what they would do, for example, is to take an animal and ligate, tie off, for example, this internal carotid artery. And as a consequence of that, that would block all the blood flow into the middle and anterior cerebral arteries on that side, producing an ischemia, a loss of blood flow, and then immediately they would give the animal a drug in order to try to keep the affected parts of the brain alive. And the general idea is that those drugs, once they were identified, would then go into clinical trials and eventually be used, for patients with this disorder. And the animal model that they particularly use in this kind of stroke research is the gerbil. The reason they use the gerbil is because the gerbil does not possess communicating arteries. It doesn't have an anterior communicating and it doesn't have a posterior communicating. It has no circle of Willis. So when they ligate or tie off the internal carotid artery on the left of a gerbil's brain, there is no possibility to shunt blood through the posterior communicating artery in order to compensate for that. And therefore the effect on the brain is a kind of pure or specific one. So if there's an MCQ in the exam, what is the animal which is most commonly used in stroke research? Is it A, gerbil, B, rat, C, hamster, or D, mouse? The answer is gerbil. There won't be, but if there was, it would be gerbil. Okay, so that's enough on the blood supplies. Just a little bit on how blood is drained out. What's the venous drainage? And in fact, that occurs in 3 stages, from capillaries that we'll see are mainly found inside the brain. And those capillaries drain into either veins which are deep in the sort of brain cavities, close to the ventral surface of the brain, or externally around the outside of the cerebral cortex. Some of those veins then drain into expanded regions. These are still veins, but they're very wide, and for that reason they're called sinuses, because a sinus is just an expanded region. And so these are really large expanded veins that you find in sort of big fissures or cavities just outside the brain tissue. And it's these venous sinuses which are encased by a double protective layer of the dura mater. I mentioned one location where we see these venous sinuses in the longitudinal fissure between the left and right cortical hemispheres, and that sinus is called the superior sagittal sinus that you find in the longitudinal fissure. I mentioned there's another double layer of dura in the tentorium cerebelli, between the occipital lobe and the cerebellum. There is a big sinus in there, it's called the transverse sinus. It's important that these venous sinuses are protected by dura, because you know a vein, the wall of a vein is just a single layer of endothelial cells. Well, that's true of these sinuses too. If they get damaged and they rupture, then there is a very strong chance that the person affected will end up with the D word. The D word is dead. All of the venous sinuses eventually drain the blood out of the skull through the pair of internal jugular veins which exit the skull through the jugular foramen. So this picture here is just a kind of indication of the network of all these expanded veins, the sinuses that you find outside the. They're shown here in this kind of bluey-green sort of picture. So look, you can see a person's skull here. This is a kind of sickle-shaped sinus here, which is found in the sickle-shaped falx cerebri between the left and right cortical hemispheres. And it's really, really huge, this sinus. It's called the superior sagittal. This one here and here, those are the two transverse sinuses that you find within the layers of the dura, forming the tentorium cerebelli. And here's, oh look, here is the cavernous sinus, where we saw that little squiggle that the internal carotid does. The cavernous sinus sits right behind your orbital cavities on the left and right sides. So you've got a cavernous sinus here and one on the other side. And here's the internal jugular vein draining the blood out of the head. There is a technique for examining whether blood is flowing properly through a person's brain, and it's called, I mean this is one of them, it's called carotid angiography. In carotid angiography, a needle is inserted into the internal carotid artery, and a contrast dye is injected into that artery, and then a simple X-ray is taken in three sequences to examine the first the arterial, second the capillary, and third the venous phases of blood flow through the brain. Angio is blood vessels and graph is looking at, isn't it? So there are three phases. Here you can see the arterial phase in the first X-ray, showing the contrast medium coming up here through the internal carotid, doing that little squiggle. These are the branches of the anterior cerebral, these are the branches of the middle cerebral that all look nice and healthy. Then you can see the capillary phase, in which the blood is very much inside the brain tissue itself, because that's where most of the capillaries are. And then a short while later, now you can see the blood that's got through the capillaries into the veins, some of these being on the outside of the cerebral cortex, and some of the blood here being in the superior sagittal and in the transverse sinuses before exiting the skull. Okay, so the final bit that we're going to look at is the cerebrospinal fluid and the ventricular system. And I'm going to take a brief pause here to take a sip of water, and you can do the same. Right, I think I'll actually show you the next slide because it shows the ventricular system, the cavities that we have inside our brains. And there are several quite large cavities inside the brain, and they're indicated here on the picture. So the ventricular system that you're seeing in the picture is kind of the ghost in the machine. It's shown here in blue. So inside each of your cerebral cortices on the left and the right sides are really large spaces which are full of CSF, and they're called the lateral ventricles. You can see that there's one here. It has a kind of anterior bit here, a posterior bit here, and an inferior bit here. So this whole thing, this gigantic cavity, is found inside your left cerebral cortex, and it's called a lateral ventricle. And there's a lateral ventricle inside your right cerebral cortex as well. The CSF, which is found within those lateral ventricles, drains into, it circulates through the system, as indicated by this arrow, into a single third ventricle, which is quite a narrow slit that you find between the left and right thalamus, you know, deeper in your cerebrum, you know, in the diencephalon, in the between brain. From the third ventricle, the CSF continues circulating down this arrow here through a narrow tube that you find in part of your brainstem, which is called the cerebral aqueduct, into another expanded space called the fourth ventricle, which is found between the underside of the cerebellum and the backside of your brainstem. That's the fourth ventricle. And from the fourth ventricle, the CSF then escapes through three holes. One is called the foramen means a hole, the foramen of Magendie, and that's found at the midline. It's a medial hole, that's Magendie. And then there are two lateral holes, which are called the foramina of Luschka. So Luschkas are lateral. And then the CSF escapes from the fourth ventricle and in fact goes on the outside of your brain into the subarachnoid space, that surrounds the entire brain surface. In fact, the CSF also passes down into the spinal cord through a narrow hole which is called the central canal, and it escapes through these foramens, Magendie and Luschka, and gets around the outside of the spinal cord as well. So your spinal cord is bathed in CSF as well. Okay, so CSF is mainly produced by a specialized network of blood vessels or capillaries and epithelial cells, which are collectively known as the choroid plexus and are found mainly inside the lateral ventricles on the left and right sides underneath your cerebral cortex. And you produce on average about 300 mils or milliliters of CSF every day. As a student said to me a couple of years ago, "Oh, that's about a Coke can, isn't it?" And I said, "Yeah." So you produce about a Coke can's worth of cerebrospinal fluid every day. As you'll see, you get rid of about a Coke can's worth as well. Now, the composition of CSF is very similar to the fluid that you find outside of cells in every other part of your body. You may recall that the fluid you find outside of cells is called interstitial fluid and is also similar in composition to lymphatic fluid. The reason for this is that the brain and spinal cord do not contain lymphatic vessels. And so one of the major functions of CSF is to substitute or replace lymphatic fluid that you'd find elsewhere in the body. And the CSF diffuses into the brain and spinal cord substance where it bathes the neurons and glial cells that you find there. It removes things that they don't want any longer, things like their metabolites, protein breakdowns, maybe toxins that might be knocking around. It removes used up and useless neurotransmitters that would otherwise float around in the brain or spinal cord tissue and might therefore cause problems with neural function. The CSF also helps to reduce the general weight of your brain by giving it a certain amount of buoyancy, and it has a kind of protective function as well because it can act as a shock absorber if you bang your head, not too hard, because the CSF is outside your brain and inside as well. So the CSF, the cerebrospinal fluid, performs a number of really, really important functions. So it's produced, and that's its composition and functions, as we saw in that previous slide that I showed you. It circulates from its major site of production by the choroid plexus, found within the lateral ventricles, through holes into the third ventricle, down a single hole, the cerebral aqueduct, into the fourth ventricle, before escaping through those foramina. You also get rid of about a Coke can of CSF every day, and that process is mediated by outpocketings of the arachnoid mater. Those outpocketings look a little bit like finger-like projections and are therefore called villi that project into the blood that you find in the one particular venous sinus, the superior sagittal sinus between the left and right cortical hemispheres. And this is basically how that is organised. Look, here's a picture I've shown you before, reminding you of the three different layers of the meninges. This is meant to be a cross-section through part of the cerebral cortex. The pink is the cortical tissue, the central nervous system containing neurons and glial cells, as indicated here. Here's part of a lateral ventricle inside the cortex containing CSF, which is able to diffuse into the brain substance. Here's the subarachnoid space on the outside of the cortex containing CSF and blood vessels. So CSF is also able to diffuse into the brain tissue from the outside as well. This is how you get rid of it. This is meant to be one of the villi, a finger-like projection of the arachnoid layer of the meninges into this blue stuff, which is the blood within the superior sagittal section. Now, as a sinus, now shown in cross-section. And bits of the CSF are picked up and into what are known as vesicles, and then deposited, dumped into the blood of the circulatory system by the regular process for vesicle extrusion, which of course is endocytosis. So that's how you get rid of CSF that's done its job, about 300 mils per day. Okay, just a little bit of pathology, because this is quite gory. There are things that can go wrong with CSF production. Actually, particularly with CSF circulation is one of the big problems. And I'll show you where there is a really sort of quite common problem associated with CSF circulation. It's right here, this narrow tube that runs through part of your brainstem. It's only a few millimeters in diameter. It's called the cerebral aqueduct. And it's taking CSF from its site of production here in the lateral ventricles down into the fourth ventricle, where it's then going to escape. But if your cerebral aqueduct gets blocked for some reason, then you end up with a situation in which CSF is being produced here in the lateral ventricles, but it can't circulate any longer. And so therefore CSF builds up in the cavities underneath your cerebral cortex, and indeed in between your left and right thalamus. If you're a fetus, in which the bones of your skull have not yet hardened, a process known as ossification. So the bones of your skull, like your frontal bone, your occipital bone, and your temporal and parietal bones, are still soft and pliable. Then the build-up of CSF inside the brain pushes the brain outwards, and it pushes those soft skull bones outwards too, producing this massively expanded head, the contents of which is mainly CSF. And that condition, a really extreme example of which is shown here in the picture, is called hydrocephalus, which means literally water on the brain. And it's a problem that occurs in fetal life when the skull bones haven't hardened or ossified, and it causes this massive expansion of the head. It pushes the brain outwards and also the flimsy skull outwards, as you can possibly guess. Children, once they're born, by caesarean section, by the way, because that head is not going down the usual canal, by caesarean section nearly always, of course, have some form of neurological deficit associated with damage to the brain as a consequence of it being compressed against the bones of the skull. But hydrocephalus, water on the brain, can also affect adults. Under those circumstances, I mean, the head isn't going to expand, but it sure as hell is going to cause quite a severe headache, because as the fluid builds up, for example, in the lateral ventricles, it does push the brain tissue outwards against the dura and outwards against the skull. And the dura contain pain receptors, and so therefore the person ends up with a headache. And actually, they will quite often come to see an optometrist, and they will say, the last few days, I've had this really, really bad headache, and my vision seems to be quite blurry. Is there anything that you can do about it? Trust me, this does happen. Because a problem with hydrocephalus, or water in the brain in adults, and the raised intracranial pressure, is that there is cerebrospinal fluid on the outside of your optic nerves, that is the cranial nerves 1 and 2. And if you look inside the eye of a person who's normal with an ophthalmoscope, you see this kind of very classic fundus image, don't you? You know, with a nice white optic disc here, and you see all the nice branches of the central retinal artery fanning out over the surface of the retina. But if somebody's got hydrocephalus and build up a fluid on the outside of the optic nerve, that causes a condition known as papillar edema, where there is water on the optic nerve head or optic papilla. And as a consequence of that, the optic nerve head looks all kind of grey and fuzzy and cloudy. It doesn't look nice and bright like this. And this is the condition that you can easily identify as being papillar edema. And if you see a person who is claiming that they have a severe headache and papillar edema of one or both eyes, then this is basically is a medical emergency. They need to be sent, off to A&E in a hospital to get to see a neurologist as quickly as possible, so that the water in their brain can be drained out, and their vision and other, and other functions can be rescued. In fact, CSF accumulates outside the brain and outside the optic nerve for a variety of reasons. Not just blockage of the cerebral aqueduct, but brain infections, brain bleeds, brain tumours, brain trauma, and really bad hypertensive crises can all produce papillar edema, in which the patient almost always complains of visual disturbances of one sort or another in the affected eye, and sometimes of double vision, because they don't see terribly well out of one eye, so they may also complain of diplopia. So papillar edema is a really important condition that you'll get the opportunity to diagnose, I'm sure, in your future careers as optometrists. Okay, so finally, sample MCQ. Which of the following statements is true? There are 2 occipital bones in the skull. The posterior cerebral artery supplies the frontal lobes of the cerebral cortex. The third ventricle occupies the space between the brainstem and the cerebellum. The dura mater is a tough protective covering for the brain and venous sinuses. Hopefully you don't have to go back over the lecture to be able to answer that question. Unlike the very, very simple MCQ that I ended the previous lecture with, this one's a little bit more difficult and could even appear in an exam. Okay, so that's it for the first 2 lectures. I hope you enjoyed them, and bye for now.